Permit me to stand on the existing protocol. I am honored and delighted to stand before my lecturers to this seminar presentation. I am Idoko Meshach Ojonoma with the math number DMS 1702083. I will be presenting on the role of probiotics in the remission of inflammatory bowel diseases. I was humbly supervised by Professor Mrs. Idoko Meshach. I'll be guided by the following outline. Inflammatory bowel disease is a heterogeneous group of chronic disease that causes inflammation of the intestine, and we have majorly two types, the Crohn's disease and the ulcerative colitis. This is a complex gastrointestinal disorder that results from an inappropriate immune response to altered intra-abdominal microbiota in a genetically susceptible host. Next slide. Figure 1 shows the type of IBD. The first class A represents the ulcerative colitis that is characterized by continuous lesion that mostly affect the large intestine. B shows the Crohn's disease that is characterized by discontinuous lesion. It affects the large and even the small intestine. Next slide. Plate 1 shows the histology of Crohn's disease. We can see in this plate, A represents the inflammation, which is a transmural one. It also represents a fissuring ulceration as indicated by the arrow. In this plate, we can also see in the B part, there is a non-quercetin granuloma. This is a representative of Crohn's disease when viewed under the microscope. Plate 2 shows the histology of ulcerative colitis. We can see in this particular plate, the inflammation is only within the mucosa region. The next slide. Symptoms of IBD include pediatric growth disorder, anemia, abdominal pain, bloody diarrhea, arthritis. Next slide. Prevalence of IBD. From studies, it has been shown that IBD is most predominant in the Northern American and also European parts of the world. Next slide. African is not left out, though we presently are noticing some incidents in this particular disease. A study that was carried out in 2020 shows about 63 cases reported in Nigeria, and we can see other countries represented in the table. Next slide. The etiology of IBD, we have interplay of factors that leads to this particular disease. While as you can see in the diagram, the environmental trigger, smoking, diet, stress, microbial flora, genetic susceptibility, and impaired immunity. Next slide. The pathogenesis of this disease, we can see in this particular diagram that the bacterial antigens are taken up by specialized M cells, which passes through these leaky epithelial cells and goes into the lamina propria through ulcerated mucosa. And after processing by the APC, that is the antigen processing cells, it is being presented to the type 1 T helper cells, which would now begin to stimulate the macrophages to release pro-inflammatory agents, which is the interleukin-1, interleukin-6, and the tumor necrosis factor alpha. And with the persistence of these chemokines is what leads to this inflammation in the intestine. Because in most of these genetically susceptible individuals, there is no regulation of the immune system, and so it continues to produce these chemokines leading to the inflammation. Next slide. Diagnosis of IBD. We have different diagnoses to help us in diagnosing this particular disease. When full blood count is carried out, it indicates anemia resulting from bleeding or malabsorption of iron, folic acid, or vitamin B12. When the platelet count is also done, you will notice a high results, which is a bowel marker, or which is a marker of chronic inflammation. Serum albumin concentration also fall as a consequence of the protein losing enteropathy, and also indicating that there is an inflammatory disease. If you carry out the ESR of the patient and also see reactive protein, you notice there is an elevation in this result due to the exacerbation of this particular disease. Next slide. Fecal calprotectin is also used as a very sensitive test that helps to differentiate irritable bowel disease from inflammatory bowel disease. In this case, sometimes the C-reactive protein may be normal, but when fecal calprotectin is carried out, it will show an elevation. And once there is an elevation in this particular test, it's indicating that there is really an inflammatory bowel disease. Next slide. We can also carry out bacterial experiments, which include the stool microscopy, culture, and examination, especially for Clostridium difficile toxin or for the ova and the cysts. We can also do endoscopy to obtain biopsy to go ahead and prepare histological slide for examination. Next slide. Radiology can be done to get an imaging of that particular, of the small intestine and the large intestine. Next slide. The treatment of IBD, there are various treatments that are used. We have the use of drugs, there's antibiotics, also corticosteroids and the rest. We also have anti-tumor necrosis factor alpha therapy that reduces the mucosal inflammation and restores intestinal permeability in IBD patients. Next slide. Intestinal microbiota-based treatment is also one of the treatments that is used in helping patients alleviate or in remitting this IBD disease, of which is the fruit of this study, because probiotics have been reported to show positive effects in this particular disease. Next slide. Probiotics as defined by the Food Agriculture Organization of the United Nations World Health Organization is or the alive microorganism that when administered in adequate amounts confer a significant health benefit on the host. Most products that contain probiotics can be in food or drugs and supplements, and even in yogurt, pickles, and cheese and chocolate. Common probiotics that are found in this preparation include the Bifidobacterium species and the Lactobacillus species. There are others, but these are the two common organisms. This is the mechanism of action of probiotics. When introduced into the GIT, we will see enhancement of the epithelial barrier, increased adhesion to intestinal mucosa. There will be an inhibition of the pathogen adhesion because probiotics are beneficial microorganisms. So their introduction into the GI tract will be competing with those pathological organisms in it. So once there is more probiotics or more life or more beneficial organisms, those pathogens will be reduced. The next slide. These are some examples of studies that were carried out to show the effect of probiotics. In this first study, Lactobacillus acidophilus LA5 and Bacteria animalis subspecies lactis BB12 was used for some patients, comparing them with other patients that were on placebo. It resulted, the results showed that the numbers of Bifidobacterium and Lactobacillus increased in both probiotic yogurt group and the control group.